All right, so now we're getting into the animal kingdom, and we're going to spend a pretty long time here. Animals are incredibly diverse. The exact number of animal phyla seems to change. It's always in the 30s generally, but people tend to collapse. Some of the phyla join them in together. Sometimes things get split apart and become new phyla. So the number generally changes. There are a number of major phyla that tend to stay the same over time. And then there's a bunch of smaller ones that most people have never even heard of that play important roles. It's very small infaunal organisms in sediment or at the bottoms of the ocean, in a variety of different sorts of places. So this isn't an animal biology course, so we're not going to go into all of them. We're really going to cover the kind of the major phyla, some of the major characteristics, and the other sorts of things would have to hold off for another time. So first off, we'll talk about what are animals really and how are they different than the other organisms we've talked about. So far we've gone through the archaea and bacteria, which are prokaryotic, and then we got into eukaryotic cells. From the eukaryotic organisms, we went into the protista, which you said is a really diverse group, and we're going to talk about some relationships between the protista and the first animals. Not in this lecture, but sort of in the next one. And then from the protista, we then went into the land plants. So from these aquatic photosynthetic protists to multicellular land plants. And then we went into the fungi, another eukaryotic, typically multicellular group, but not always. Some members were not. And now we're going to go into the animals. So the animals are going to be based on some of those other characteristics and traits that we mentioned. First off, eukaryotic. All right, so they're all going to have eukaryotic cells. They are multicellular. Right, so all animals are multicellular. There are no single-celled animals. So we have some protists that are sometimes referred to as the protozoa, which are the animal-like protists. So they're single-celled organisms that are heterotrophic, which is going to be our next thing here. Heterotrophic meaning they obtain their nutrients by essentially eating other organisms or living things. And typically the digestion will take place within some sort of chamber within the animal itself, which is a multicellular organism. Now, we'll see some variation to this when we talk about the sponges, for example. The sponges are going to be the first group of animals we talk about. They are eukaryotic and multicellular, but they do not have a digestive cavity. Their digestion occurs at the cellular level. So it's like as if they're a colony of protists that kind of work together to carry out a variety of different functions, right? And except they are animals, or they're not protists, but they're the sort of the first step in that. But they're all heterotrophic. Now, some animals have photosynthesis occur within them because they have associations, just like some of the fungi have associations with algae, and the algae do photosynthesis, and the fungi benefit from that. There are animals like this is sort of like an anemone here, or like a polyp that could be part of a coral, and the corals in particular, their polyps have symbiotic algae called zooxanthellae that live with them, and they're essential, the relationship between one and the other. But that does not make the animal photosynthetic. They have a relationship with a photosynthetic organism. The animal itself is heterotrophic. So all the animals are heterotrophic. Now they're going to lack cell walls. So you can say, how is missing something an actual characteristic of a group? Well, it separates them apart from the other groups. So the fungi had cell walls of chitin and a variety of other materials. The plants had cell walls of cellulose. Right? So we have different groups of multicellular organisms, multicellular eukaryotes, that have cell walls, and they have cell walls that are made up of different material, but animals have none, no cells. They sometimes have an extracellular matrix that the cells are within, but that's something sort of separate. Now, other things that people often talk about with animals, about them having behavior and being able to move, those things are not necessarily true for all animals. There are many animals that are sessile or sedentary, and they do not move. They're essentially attached, fixed to one spot. and never move from that particular location. Most of them still have some ability to move within themselves, but some don't necessarily. They move cilia and or flagella that they have lining their cells, but the organism itself doesn't move. So we'll get into some of those things, but generally there's movement in animals, and we're going to have different types of tissues. So the first group of animals we talk about aren't going to have these tissues, but generally most animal groups have unique tissue types and many cell types. So this is one of the big things that we're going to find with the animals, with the fungi and the plants. There are fungi few different types of cells, really comparatively. With the plants, there are a few more different types of cells and some more different types of tissues. But with the animals, we're going to see a great diversity in tissue and cell types. Really a lot of diversity with a whole variety of different sorts of jobs for all these different cells. We're not, again, in this particular class, we're not going to get to go into all of them, but we're going to bring up a lot of the major ones and major groups, and some of them soon. Right now, in this particular lecture, as we talk about how they form through development. Okay, so really kind of highlighting something that's true for most all the animals. Eukaryotic cells, they're multicellular and they're heterotrophic, and the lacking of the cell walls are kind of like the core thing. One of the hard things about animals, and then we talk about some of the groups, is that there's always going to be exceptions. So there's going to be, all the animals have this characteristic, and then we'll say, except this group that doesn't have that sort of thing, and that will be a common theme, that there'll always be something coming up that's different, but we're paying attention to the big picture and the main core characteristics. Okay, so let's talk first about animals, how they kind of come about, and we'll talk about reproduction in animals really first, just very briefly, and we'll go into it more detail with different groups. So for some of the other groups we talked about, one of the first things we went into is sort of a life cycle for those organisms. That's not necessarily something we could easily do with animals, because again, there's a great amount of diversity within animals. So first we'll talk about what are some of the big picture differences that they have. So one would be with reproduction. And first, we'll talk a little bit about, just briefly, asexual reproduction in animals. So asexual reproduction in animals can occur in a couple of different ways. All right. So this is where we do not have mates. Right. So one individual gives rise to more individuals. Now, so typically, the individuals that are given rise to are going to be clones of the original, which means there's a lack of genetic diversity. If the organisms are living in an environment that suits them well, where there are a lot of nutrients and there's not under stress, producing a large number of individuals quickly without having to find mates and store up all the nutrients that they need to develop gonads and gametes and all these other sorts of things is an advantage. All right, so that's a big advantage to asexual reproduction, the speed of it and the lack of complexity of finding partners and the developmental aspect of it. The drawback of asexual reproduction is that because all the individuals in that group are going to be exactly the same, there's no genetic diversity. So if something changes in the environment, that population won't necessarily be able to adapt, and all the individuals might be wiped out, as opposed to a different group that has some genetic diversity in it because of sexual reproduction. Then some of those individuals may survive the changing environment and be able to adapt over time to that new thing. So that's the advantage, disadvantage of asexual reproduction. Mechanism-wise, we're not going to go into details, but how generally does this occur? There are a couple different ways. One of the most simplistic ways that would occur is something called fragmentation. So, kind of define there in part of the term, they're fragments, right? So pieces of the organism, and this is often more so happening because of something in the environment. So something, say, cuts off a piece of this organism, a fragment, of it. And so that fragment of the organism then falls to the substrate, and then perhaps it has the ability to attach. The cells in it can reorganize themselves, start to express genes to form new structures, and this little fragment of this organism may start to develop into a whole new organism. So sort of a new clone organism. But again, this would be a broken off piece, not something the organism did on purpose themselves. Not all animals can do that. Many cannot, but there are some that can. All right, take a piece of them and actually regrow that piece into a whole new animal. And we'll see that among different phyla as well. There isn't just one phyla that can happen. It can happen in a number of different phyla. So that's called fragmentation. We then have something that's a little more purposeful, right? Something called budding. So we see budding in some groups where, I'll do this in a different color so to kind of note that it's something unique. So on the side of this organism, a little bud begins to form. So it's like a small version of the organism itself. Now there are some animals that we'll talk about that are called colonial animals. So essentially so organisms like this as well, the cnidarians, there's a polyp. All right, so this polyp is the whole entire organism itself. This would be like the mouth here, this would be the digestive cavity here, and then as a little bud grows off, there's actually a connection between that bud's mouth and its digestive tract and this digestive tract, and they actually form one whole organism. And as this happens again and again and again, they're a colony. So there are actually many polyps all making up then one organism. Each polyp is sort of a clone of the others, but collectively they're one organism. In this particular case that we're talking about with reproduction, that means somehow it separates. So the organism creates the bud, and then at some point it breaks it off, and then that little budded organism leaves and then forms again a whole new organism that's a clone, and that would be produced by budding, and that would be something on purpose. The organism did that through gene expression and control and regulation based on certain signals and timing, and it's programmed to actually do that, as opposed to the fragmentation, which was just something snipped off or cut off. Another animal ripped it off, or a rock crushed it and broke a piece off of it, and that piece either just degrades and dies, or it actually grows into another organism. And then we have fission. Which is a bit more dramatic, but we do see this in some organisms as well. It's where the organism itself sort of rips itself in half. And so what goes on here is the organism starts to split essentially down the middle, and it tears itself into two pieces like this, and then each piece is essentially half of the original organism split down the middle. Again, they're both clones but it's not like one is smaller than the other. They're fairly equal in size. That's sort of the main difference there. So in fission, the organism essentially rips itself in half, and then each half becomes a whole new independent organism. But again, they're genetically identical to one another, and they are clones. Right? So these are some types of sexual reproduction. We also see some types of asexual reproduction. And then there's another type where we start to go down the sexual reproduction path where gametes are formed. For example, so an egg is formed. In many particular cases, the ova doesn't complete meiosis until it reaches a certain signal. In some organisms, the meiosis process doesn't complete until the sperm actually begins the fertilization process, and then it completes meiosis. So essentially what I'm saying is some ova, the unfertilized eggs, are not 1N or haploid right at the time of fertilization. Some of them are actually 2N, and then they complete it, which means that in some organisms, even without fertilization, that ova could start to divide and develop into a 2N diploid organism, and that does happen in some cases. So the egg itself, unfertilized, just starts to divide and form a whole new individual. That is technically asexual reproduction, but there's sort of a sexual aspect to it in that meiosis does occur, so there is recombination of genes, and we can have some genetic diversity then among the offspring. So even though the offspring were not produced by fertilization, they so they're asexually reproduced, but they can be different from one another. In these examples I'm talking about here, they're all identical. They're identical organisms, exactly the same as one another. There's no difference. Okay, so that's in asexual reproduction. And I'll talk a little bit more about the parthenogenesis and this aspect of the eggs developing without fertilization later on as we talk about the different groups who actually display those characteristics and traits. So we'll get to that. So for sexual reproduction, in animals, most of the time, they're males and females. But there are a number of organisms that are hermaphroditic. Organisms that are hermaphroditic display hermaphroditism in also a variety of different ways. So some, for example, are simultaneous hermaphrodites. That means they are a mature male and a mature female at the exact same time. And so what happens in those populations is that two individuals just find each other. It doesn't matter because they're all exactly the same. And then one individual transfers sperm to the other. That one transfers sperm to the other. They fertilize each other's eggs, and then they start to develop new offspring. And they're all unique individuals that are produced by sexual reproduction, but there's not the separation of male and female. Some are called sequential hermaphrodites, where at one point in time they're male and then they switch and become female, and there's others that vice versa that are female and then become male. So this happens in invertebrate animals and invertebrate animals, as well. So we have among the animal phyla, there are many examples of sex switching and hermaphroditism in a variety of different sorts of ways. So again, I'll bring that up as we talk about reproduction in some specific groups to give you a few of those examples of when that particularly happens. Because of this, what we do sometimes see is in the organisms that have males and females, there's often differences in or among the sexes. So one group, and this also varies, might be much larger than others. Actually, in many animals, the female is much larger than the male, partly because she needs a lot more nutrients stored to produce the eggs, which are much larger than the sperm. And also the female, if they happen to hold on to the embryos and protect them during development, again, needs space for them to develop and often nutrients for them to give to the embryos. In addition, sometimes that they won't feed during that particular time, so they have to have their own stored nutrients. So in many cases, and many times that's the case, but not always. Sometimes it's the other. Some, as they compete for mates, the female displays to the male, but again, in most cases, it's the male that is much more colorful and has to do a lot of different behavioral things to attract female mates. Females usually don't have to attract mates. It's the males that usually have to work, sort of to do that. Again, and that's going to be very dependent on different groups that we kind of talk about, but these are just some of the generalizations. So, what is common among all these different sorts of things that do all these things in different ways? Well, generally the males will produce sperm, and that's going to be fairly uniform and the same type, of structures among all different sorts of animals, and the female produce an ova or an unfertilized egg. And they're both going to be haploid structures. As I mentioned, sometimes the ova isn't quite completed with meiosis, and I'm going to erase some of this because I'm going to use some of the area here in a minute to do other sorts of things, or there's triggers that will cause the meiosis to complete really before then the fertilization process begins. But what will happen then is that once fertilization occurs, you have a zygote. All right, So this is a two-end cell, and that's the product of the union of the sperm and the egg. So this is the beginnings of our embryo. So this is the product of fertilization. And it's going to be genetically unique, the combination of the unique sperm produced by meiosis, the unique egg produced by meiosis, and then they come together. And again, millions of different sperm with all different combinations could fertilize that egg. So all the offspring are going to be completely different from one another. Well, not completely, but they're going to be different. They're going to be different from one another and have a number of genetic traits that might be unique. What will happen, though, again, as a common sort of thing, is the cell then is going to divide. Right? The division process in animals we refer to as cleavage. And that's because there's actually a thing called a cleavage furrow, where internally part of the cytoskeleton that interacts with the membrane is going to start to tighten and sort of pull. It's going to squeeze sort of the cell, and the cell is going to kind of pull together toward the center until it divides into two cells. So it's our mitosis process, but we call this cleavage because there's going to be certain patterns to the cell division. There are a number of different ways. Two of the more common ones are called spiral cleavage and radial cleavage. So, and this will depend on, as these divisions occur, sort of the pattern that the cells divide, when they stack on top of one another perfectly, or do they start to, and the next time, kind of fill in, in between the other cells, and they're kind of rotating around. Again, these are different patterns seen in different organisms, and as we watch their embryonic development, it tells us something unique about them. These are traits genetically programmed, so that's stored within the genes of the organism. So when we see organisms that do the same thing, that means they share some of the same genes, and when organisms do something different, that means they have different genes dictating a different sort of process to occur. But generally we have this process that eventually results in sort of a ball of cells. And this ball of cells that we see in most all animals, I mean everything from really, you see when we talk about the first group of sponges, which are not necessarily even true animals because they don't have true tissues, to cnidarians, to humans, to everything, in between, invertebrates, worms, and snakes, and birds, and everything in between typically forms some cleavage pattern, and then a blastula, a ball of cells. The blastula, what happens is that some of the cells in the interior start to secrete molecules right toward the very inside. So there's differentiation. So cells on the outside are starting to express different genes, and cells on the inside, so the center of the ball, are expressing different genes. And these cells start to secrete chemicals that bring in water. And what occurs is then sort of a hollowing out So there's actually a chamber then in the center here, water and other chemicals that were secreted by these cells, and this hollow now ball of cells. So I'll write this is called a blastula. And this chamber inside here is called a blastocell. So we're going to see this a lot, C-O-E-L. Cell means cavity. Okay, so I'll write this over here because you're going to see it a lot with animals. We'll talk about groups that are acoelomates and pseudocoelomates and the true coelomates and coeloms formed by different processes, the schizocele and intracele. So it's all talking about cavities. Okay, so some kind of cavity inside an animal. So you have, one cavity that runs through your body. It's your digestive tract. Okay, mouth all the way through, or your intestines. Okay. You have a second cavity inside your body. That's the space in your body where you have your organs. So your lungs are in that chamber or cavity. Your heart is in there and your other organs. So there's an outer cavity, and then there's this inner cavity, a tube essentially running through it. Many animals are then constructed, their body in this way. They have the digestive cavity, and then they may have another secondary cavity outside it. So these are all called coelums. In this particular case, this is called the Blastocell, which is an embryonic structure that all animals are going to have. Now, again, we're talking about pretty much everything from, again, the Cnidarians to us. Right? What will happen with almost all animals, again, differential gene expression in cells right here at this lip. Okay, you got to remember this is a three-dimensional ball, so this is kind of like a little tube being pushed. So if you had a balloon and you're pushing your finger in the balloon, and a little kind of chamber is then forming as you push your finger inside, that's what this sort of looks like. So these cells are actually moving inward like this, and these cells start to differentiate. So they actually start to become different types of cells, different than the other cells. These cells on the outside are called now a germ layer. So these are embryonic cells that start to form embryonic tissues, and the undifferentiated tissue, or partly differentiated now, is called ectoderm. All right, so ectoderm. This ectodermal embryonic layer is going to give rise to typically skin and the nervous system. This group of cells that start to move inward here, like this, hold on. They are going to be called endoderm. So endoderm versus ectoderm. The ectoderm, again, I'll just say skin, but other things too. But it's just kind of the outside of the organism, whereas endoderm is typically going to form the gut, right, or the digestive tract of the organism. So in some organisms, this sort of opening to that tube will become the mouth, and then this little chamber will become its digestive tract. That is going to be true for, again, most all organisms. Now, in some it flips around, and so the first opening is the mouth, and then the second opening, if the tube continues all the way through, would be the anus. And others, it's vice versa. In others, the first opening is the anus, and the second opening is the mouth. But for the organisms that we're going to talk about in the sequence that we're going to cover them, we're going to start off with organisms like this that has a blind gut, which essentially means that here there's a mouth, and then this compartment here is its gut or digestive tract. It doesn't continue. There's no other opening. There's only one opening. Right? So this is called often an incomplete digestive system. Where food comes in through the mouth, it goes into the gut, it's digested, broken down, nutrients are absorbed, heterotrophic organism. And then any waste, any undigested material, all that stuff actually comes back out through the mouth. So the mouth has food coming in and waste going out the same opening. And we'll see that in a few different groups of animals, a few different phyla of animals. We'll have that incomplete gut. And that all forms through this process that is called gastrulation. So gastrulation is incredibly important process in the development of animals. It is the process that not only begins to develop their digestive system, which is a key for these multicellular heterotrophic organisms, but it also begins the differentiation of tissue types, meaning that you start to get different types of tissues, ectoderm, endoderm forming, and then in some groups of animals, so we're going to have animals that are called diploblastic, so di meaning two, and they're just going to have these two layers, endo and ectoderm. That's it. But then in other groups of animals, a third, and most of them really, a third tissue type is going to form in between the two, and they will be then called triploblastic. So obviously for three. So all animals are going to be, except sponges, either diploblastic or triploblastic. Okay, and then the triploblastic ones will have this extra group of cells that form a type of tissue called mesoderm, which we'll talk about again later, because the first couple groups aren't going to have this. So we'll talk about it more when we get to those groups. The mesoderm will become things like your organs, bone, blood, and all these sorts of things. So the first many groups of organisms you're going to have are not going to have those. The mesoderm also become muscle tissue, as well. And so in organisms like the cnidarians, which is a group that has like the sea anemones and the corals, while they have movement and muscle-like activity, they have no true muscle because they have no mesoderm. So to form a certain type of tissue, you're going to also have to have what comes before it, the earlier previous tissue type. And so in this particular case, the ectoderm give rise, say, to the skin and epidermis and those other related structures. The endoderm will give rise to digestive cavity, and then if it has different chambers and all different parts to it, but they're necessary, you have to have endoderm. And then if you're going to have bone or muscle, you have to have mesoderm. If you don't have mesoderm, then you can't have anything that's truly one of those tissue types, because it has to develop from them first. So, key things to get out of this. We're going to stop here and then start to get into the first animals, which would be the porifera. And then we'll get into the formation of them and then some relation to the protists. But from this, animals. What's an animal? Multicellular, eukaryotic, heterotrophic. Okay, so you have to be multicellular with eukaryotic cells. They're heterotrophic. They have to ingest food and then digest that food inside either their individual cell, cellular digestion, or usually within some kind of gut cavity itself. Because of that, most animals are going to have tissues, right? And we'll define that later specifically, like what is a tissue truly? Like what's the definition of it? What do we need for it to be called a tissue? Tissues are going to be made up of the same type of cell, and then we're going to have all different kinds of cells in animals that are going to make many different types of tissues. Though all those tissues come from embryonic original tissues. The way they form is that a fertilized egg through sexual reproduction starts to divide through a process called cleavage. It may have a particular unique pattern depending on the group of animals. It forms this little ball of cells that hollows out. The hollow ball of cells is called a blastula. The inner cavity, the inner chamber, the liquid fluid-filled cavity is called a blastocell. Cells toward the bottom of this cavity start to express different genes that then pull and move the cells inward, and they start to then form this opening and chamber, which will become the digestive tract. Those cells are now, and those tissues are now going to be called endodermal, and then eventually give rise to some type of digestive system. Right? If it stops there and that's all there is, it's just a diploblastic organism with two tissue types. If it continues and then more develops, which we'll get to again further on, it'll be a triploblastic and have three. Now, an adult organism can have many, many, many different types of tissues. These are only referring to embryonic tissues. Okay, so when we talk about diploblastic and triploblastic, these are the origin, the original tissues developing as the organism just develops from an embryo. Okay, and then the asexual reproduction part, talked about a couple different strategies that some have. Some are somewhat accidental or environmentally induced, pieces being broken off of an organism. For example, say sea stars can do this as well. Some people on the Great Barrier Reef found that there were certain sea stars that were eating corals and destroying the reef, and the reef was the ground for the young fish to develop. So when the corals were destroyed, then the fish were gone, and so their fishing industry was a collapse. So they went in to say, let's kill all the sea stars so we can get the corals back and get all the fish back. So they collected up sea stars, chopped them into tiny little pieces, and then just threw the pieces back overboard, and good riddance to them. Except a short while later, the reefs had 10 times more sea stars than they did before. So what happened? How could you collect all the sea stars, chop them up and destroy them, and get more sea stars? Where did they come from? All those little pieces. So by chopping them up into pieces, each little piece became a whole new sea star. All right, fragmentation. Okay, so the sea stars didn't do this to themselves. People did it to them, but they were able to regrow whole new individual animals from just their parts and pieces that were cut up. Those would be clones and genetically unique, or genetically identical to one another, at least the ones that came from an individual. Some can do it themselves, so they actually will bud off a little piece, break it off, and then that becomes a whole new individual. And then some actually just rip themselves in half, a process called fission, where they pull themselves apart. So those are due to environmental conditions and a variety of things that might trigger an organism to do that. So that's kind of the first part of an introduction. One term here I'm missing here is we said the process was called gastrulation, so the structure itself that has this little tube forming in it would be called a gastrula, just so that I don't miss that. All right, so next thing will be sponges.